This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hello, this is Audiommunity, recorded on July 10th, 2025. I'm Kevin Bonham in Boston, Massachusetts, joined by Matthew Woodruff down in Atlanta. How's it going, Matt?
Hey, everybody. Doing okay. Recovering. Those of you that have been following along may notice that there was a slight gap in recording.
It's true.
Part of that was because I was ill. And I have to say that today is probably the first day since I canceled on that illness that I would have been able to physically do this, because I have been coughing my brains out for 2 weeks, like wheezing, awful, terrible, post-viral, just like not good. So
awesome.
I'm
COVID or flu or any knowledge of what it was?
I tested for COVID and flu early on in the process, and they were both negative. And I think that it is a liar. I believe that I had COVID.
I wonder, do we know how good the at-home antigen tests are for the latest variants? I remember there was a lot of discussion around this, like around Omicron, and they were saying, they're still good, but gotta imagine that they're getting less and less good.
Yeah, and it's not clear that they all continue to infect in the same places, right? So, like COVID, an interesting question early on was, is a nasal swab good?
Right.
Should it be a throat swab? Is that going to give you reasonable? And I would say that the most recent variant I've heard by the medical field is called throat raisers. And I would say throat raisers, not a terrible descriptor of that. And it makes me wonder if the nasal swab that I was doing captured effectively what was going on. But
well, that would be a cool clinical trial to do if anyone was getting any money for COVID research anymore. Yeah, but I suspect they're not, at least in this country.
They are not. So. And so to answer your question, we almost certainly don't know how effective those antigen tests are. I would say that the thing that the closest thing to a consortium that would answer those tests that I'm aware of was shut down because the pandemic is over. So
yeah, good. Well, if it makes you feel any better, my family encountered hand, foot, and mouth disease this past week.
That's the worst.
So, and for my kids, it turned out to be hand, foot, mouth, and butt disease.
Yeah.
Mostly okay. I mean, Rachel and I didn't get it, so it was just loss of childcare. But the kids were miserable for a few days.
Yeah, that one's bad. Fortunately, I mean, most of us get that one as kids, and you are pretty much protected forever. Emily got it. As an adult, having not gotten it as a child, and we were on vacation that week, and I have to tell you, that was the most miserable I've ever seen her. She was just covered in ice in a bed covered in ice. It was horrible. Ugh. It was absolutely horrible. So I'm sorry to hear that. Unfortunately, butt involvement is not an uncommon feature of that.
Yeah. Well. There is no good segue between that and the thing we're going to discuss today.
There's not.
But that's fine.
No.
Hopefully, those of you that are listening along will forgive us this. We're going to do a paper today that I found shared on Blue Sky. The title of which was a little bit, I don't know why, off-putting, but I'll just say it. So the subfornical organ is a nucleus for gut-derived T cells that regulate behavior. And the first author is Tomomi Yoshida. And it looks like there's co-corresponding authors, David Hafler and Andrew Wang, from Yale, I think. Yeah. So I don't know why subfornical makes me think of something like somewhat unseemly, but it turns out that this is a brain region. The fornix is part of the brain. And so I think we're going to talk about all of that. You seemed like you had something you wanted to say.
No, I was going to say, I think it's the combination of subfornical and organ, actually. I think either one of those two words separately is fine. I think it's the phrase subfornical organ that for some reason is like an alien abduction.
Yeah.
Or
it's like related to fornication somehow, or I don't know. Like there's some... Anyway, so I thought... the reason that I picked this paper, I feel like we've got a new theme here in season 2 of Audiommunity, a few themes, one of which is this idea of location, and another of which is a theme of the immune system being in places where people didn't previously expect it, or being affected by location in ways that people didn't expect. So the whole premise of this paper is essentially pushing against a concept, again, we've talked about a bunch, even in the last few episodes, about the blood-brain barrier, this idea that the brain is this quote-unquote immune-privileged zone where the immune system doesn't go, doesn't reside.
Lies.
That was always suspect, I think, but I think more and more evidence is showing that that's just not a thing.
Yeah.
And I think this paper does a really good job of sort of demonstrating the importance of these T cells that are located in the brain in this subfornical organ, and it has a link to the gut microbiome, which of course appeals to me as well. And I feel like there's this really interesting something that I'm getting more and more interested about is the idea that a ton of our physiology is built around this idea of interoception. So it's a sense that we don't really have a ton of access to, like we don't think about it a lot, but our body works really hard, including our brain, to understand its internal state. And a lot of that information doesn't come up to consciousness, but it still matters in terms of regulation of a bunch of functions. So anyway, that's sort of a high-level overview of why I picked out this paper. What did you think of it?
Yeah, I mean, I think you touched on a lot of important points. I always, and we've touched on this before, but every time I read a paper like this, I remember back in grad school early on when somebody was talking about immunology, and they were lamenting that all of the easy stuff had been done. Like all the subsets had already been discovered, and how easy a time it was that everybody had it, and that there wasn't really anything left to do. And I don't know why that stuck in my brain, because I do actually, because I think at the time I was like, that seems silly. I hope that that's not true. And then you read papers where you're like, we found CD4 T cells in the brain because we looked there.
Right.
Like,
yeah.
Okay. Cool.
Yeah.
And no, I think it goes exactly to what we've been talking about, which is that immune privilege is not the absence of immunity. It simply isn't. It is a careful monitoring of the site and a very selective way that cells respond within that environment, right? And that doesn't mean that the concept is wrong. It doesn't mean that, there is no such thing as immune privilege in the eye, for example. It just means that privilege doesn't exist because there is a complete absence. It's that there is selective tolerance and selective response protocols, basically. In
Philosophical question.
Sure.
Then.
Yeah.
Is there any tissue other than like the blood that doesn't fit that description?
Not really, no. It's just tuned, right? It's simply tuned to the site. Your immune system sees everything, and it's tuned to the site. Now, there are places certainly where an absence of sensing is part of, or the restricted nature of sensing is a part of that, right? So you could make an argument that still in the eye, right, you've got a very careful way that things are sensed. But no, I think that we're talking about grays here now. We're not talking about black and white. And that was always the way that immune privilege was discussed. You have tissues that are, and you have tissues that aren't. And I don't know that that framework continues to be useful.
Yeah, I think, this is another theme maybe of season 2. I think I've mentioned this idea before, this concept that, like, it's most familiar to me from Buddhism, but this idea that, like, whatever concept, like, we sort of reify these concepts, like immune privilege, and we think that there are things that are immune privileged and there are things that are not immune privileged. Or, we think of a cell, it's either an antigen-presenting cell or it's not an antigen-presenting cell. Like, there are lots of gradations in those states, and, I think immunologists in particular are very enamored of this idea of boxing things, like drawing neat little, you might almost call it a fax gate around particular things. And I think more and more it's clear that there are really interesting discoveries that are to be had if you recall that these definitions, these paradigms that we learned in grad school they're just models of the world, and they're not necessarily strict.
Yep.
So with that, let's get into the paper. I thought it would be worth talking about a few sort of definition things that come up in the introduction and in the first few things that I had to go look up.
Sure.
So one is, first of all, the fornix. So this is the fornix itself is a bundle of nerves. I wrote down that connects the... Oh, did I write that on the next page? I wrote my notes somewhere. I think it connects the amygdala to another section of the brain. But the fornix itself is not so relevant. It's just that locates then the subfornical organ. And the subfornical organ is underneath the fornix. That's where its name comes from. But it's part of... it's called a circumventricular organ, of which there are a few of these in the brain. And so circum, around, ventricle, so this is like around the spaces of the brain. There are these different regions that are basically infused with a lot of blood vessels. They're getting a lot of signal from circulation. And the SFO is one of these regions. And in particular, it's very vascularized. So that means there's a lot of access to circulating immune cells, and the possibility, therefore, as we'll see, for the immune system to sort of interrogate this space and have cells reside in that tissue a little bit more durably. It's part of, apparently there's these layers called the meninges around the central nervous system, so around the spinal cord and the brain, and there are 3 layers of them. The outer layer is called the dura, and that's where this SFO is located. So that's just, these things came up a bunch. And the other phrase that I thought was interesting, I've talked about this seemingly knowledgeably. I think I've read it tons, and I didn't actually make the link. So it's just this idea of the parenchyma versus stroma. And so stroma, you kind of know, it's like the structure of a tissue or whatever. The parenchyma is just like the functional bits. So when we're talking about the central nervous system, parenchyma would be like the neurons and the glia and the cells that are actually doing the function, whereas the stroma is just like the things that form the structure of the organ, and they're not necessarily strictly critical for that organ's specific function.
Right.
So,
which again is a nice example of bins
yes.
we like to place on things. I love relational definitions that. It comes up in immunology all the time, like all the lymph node structure and function stuff. We like to put the box of things that are doing things and the box of things that aren't doing things, and you're like, but the two.
Yeah.
They're doing it together.
Right.
Right. But yeah, sounds good. So we will go with those definitions, and I'm going to promptly leave them all behind because I am not a neurologist or a brain biologist, and it's going to immediately escape me. So we're going to stick with subfornical organ. We're going to point to Figure 1 and say, if you are interested in detailed structures of the brain, they do a good job of pointing out many of the things that Kevin just said in Figure 1B. I would highly recommend that you start or you take a look at that. But in particular, take a look at the middle top panel of Figure 1B, and you get a nice little highlight of where that subfornical organ actually resides. And so that is what we will be...
In a mouse brain.
In a mouse brain. That will be largely what we'll be talking about today. And I was not kidding when I said that the fundamental observation in this paper is that there are T cells in the brain. And so you start that, of course, by looking for T cells. Figure 1A is a good start for any graduate student rotating in a lab, which is why don't you go stain a brain for CD3 and CD4? Of course, CD3 is the signaling component of the T cell receptor, so that should give you T cells, and then CD4, specifically your CD4 positive T cells. So the question is, are they there? The answer, of course, is yes. We found them. And so, you have off to the right there in C, basically look at how dense are these different parts of the brain in CD4 T cells. And so two different locations show up. We will not talk about the choroid plexus, but the SFO next to it. You can see that the density of T cells in those images is actually pretty high in comparison to these other spaces.
And it's worth noting they put in a lot of effort. I think one of the things that they really have to do, and they do, is to spend a lot of time convincing people that this isn't like we're sloppy with our surgeries, we're sloppy with our dissections of these regions, and we just got contamination from like blood vessels and stuff like that. They're really trying to say, actually, these are not T cells that just like happen to be in the same general vicinity we're pulling out. Actually, these are residing in this specific location.
Yeah, and they'll do a couple good experimental things in the next figure that'll really drive that home. So they take a look at for this reason in D, they take a look at these brain sections in a slightly more three-dimensional way, because one of the things that one could say is, well, you're just getting a projection of fluorescence, and you can't actually tell if those cells are in the blood vessel or if they're not in the blood vessel, right? And so one way is, or to combat that kind of thing, is to take a thick section of the tissue and do a really careful three-dimensional reconstruction and say, well, yeah, but if you really look carefully at this blood vessel in 3D, right, like you actually don't see the T cells in there. They're outside of that space. And so they do that in D. And I apologize, periodically I'm just going to cough up a lung. And I think what drove this paper in my mind from an interesting set of observations to a Nature paper is that they do a really good job of periodically taking what they're finding in the mouse and moving into a really complicated human system.
Yeah.
And this is we didn't do what's good in 2025 this morning. And I won't go into too much, but one of the things that has happened recently is the NIH has announced that it appears not to be interested in mouse modeling anymore, or at least insofar as doing only mouse modeling. Like it appears that we're shifting towards you must be doing translational biology or it's not worth it sort of thing, which is dumb. But it is relevant in studies like this to go back to human systems and make sure that we're not chasing our tails down rabbit holes. I'm going to mix all the metaphors. So
talk about mouse burrows, chasing our tails down mouse burrows.
There we go. So in E, they do what you'd hope would show some sort of translational relevance was they actually find donors. So these are post-mortem human brain samples without neurologic disease, importantly. And basically what they're showing is that if you look in the SFO of a human brain section, you actually do see a similar increase in the number of T cells in that density. So some indication that what we're seeing in the mice here at the top of figure 1 might be relevant to humans. And then you do what you always do when you find a new cell in a new space, which is
Single cell
transcriptomics, right? And so what we're doing here is we are taking T cells from 2 locations, from the brain formally and from the meninges, which is sort of outside that brain space. And we're asking the question, are the T cells that are formally in the brain that we're collecting from the SFO, basically, or surrounding areas, are those T cells different in some way, specialized in some way, that might indicate that this is not just a circulating T cell population? This might be a meaningful T cell population. And the answer seems to be yes, there are differences. When you look at the UMAP that they come up with, you find that those T cells that are isolated from the brain versus the meninges do seem to cluster off by themselves a little bit. And some indication, although I think they might overplay their hand a little bit here, some indication that the T cells that they're pulling from the brain appear to have a more active or differentiated phenotype. So certainly things like CXCR6 are up. That'll come back again. CD69, which is an activated phenotype, it's not clear what it means here. CXCR3, and then maybe some of the Th1 polarizing cytokines, right? So interferon gamma, TBET, TBX21, probably all up. Now they also make a case for some of the Th17, Th2, Treg kinds of things. Maybe. But certainly that Th1 cassette, which they then highlight, and they pull out essentially off to the side there. So I would say, some good indication that you have a specialized T cell population or a more differentiated or different T cell population in the brain than in the blood, essentially.
And yeah, I thought, I was actually surprised at how well segregated the T cells from the brain and the meninges were in this, because the meninges, if I understand correctly, the way that they're doing these dissections, essentially they are. So first, I want to highlight they're doing something called intracardiac perfusion. So, and I had to do this once when I was rotating in graduate school. So you take a mouse. You anesthetize it so it's unconscious. You open up its chest cavity, and then you have a large needle with a pump with a fixative that's going to sort of like freeze all of the tissue in place. And you would put that needle directly into a ventricle of the heart so that it immediately gets pumped and completely perfuses the animal with this fixative. And so once they do that, now they've got basically everything locked in place, because of course that's getting into all the capillaries, it's getting into every single tissue, everywhere where blood goes, which is everywhere, is now being fixed. And then they separate out the membrane that surrounds the central nervous system, and I think that's going to include all of that vasculature, right? And then separately they can take the brain that is not vascularized, and then that's where these other T cells are coming from. So it's very, very clear from the UMAP of the single cell transcriptomics that the T cells that are coming from the brain just have a almost diametrically opposed signature. They're you can sort of draw a line down the middle of that clustering, and it seems very clear to me. The separation that's happening.
No doubt. No, I think you're absolutely right. I think definitely some indication that that there's something interesting going on in these brain resonant T cells, right? So, what are they? What are those T cells actually doing? And so, this gets to, we get into figure 2. And we now want to know, and you'll notice that my voice has changed because this is what's been going on for the last 2 weeks. So I may or may not be able to talk through figure 2. Hold on. How do you know that a T cell is not coming from the blood? And the answer is actually there's some fairly straightforward immunologic techniques that allow you to do that. So one of the things that we can do when we're investigating this and understanding what these T cells are actually about is by doing... Hold on. Yeah. Not great. Not.
If you need me to take over, let me know.
Yeah. Why don't you start talking your way through figure 2, and then I will catch back in. But what I was going to talk about, if I can get it out, is the fact that what you can do is you can take an antibody. Nope, it's not going to happen. Go for it.
It's fine. I'll just back up for a sec and say in the one of the things that they point out in figure 1 about this different signature that's particularly relevant is they find CXCR6, which they're saying is this is a marker of tissue residency. It's essentially something that is upregulated by T cells that helps them to enter or stay in tissues. Then you typically don't see this in blood-derived T cells. So that's like an extra bit of evidence. Now, the first sentence of the next section made me kind of roll my eyes a little bit, which was that so they do a tissue screen. So they basically are looking for this this gene signature in a bunch of different tissues. And they say, in our tissue screen, we noticed that white fat T cells also had a high frequency of interferon gamma positive T cells. Now, you have to put this line in your paper, but like they were looking for this, right? Like they knew what they were looking for. This feels like a sort of very post hoc, like, oh, what do you know? We happened to see the signature, but it is interesting that of the tissues they looked in, they pulled out T cells, they looked at gene signatures, and the tissue that had the most similar signature to these SFO T cells were from white fat.
Yep.
And so now they want to claim, like, I hate this phrase axis. There's the gut-brain axis. There's the gut-brain microbiome axis. They want to claim there is a fat brain T cell axis.
Yes.
Okay. But basically what they're going to do now is they're going to label the circulating T cells so that they can basically exclude those from their tissue profiling. So they're going to... Oh, go ahead.
No, I'm agreeing with you. Labeling.
So they're going to explicitly label the cells. So this is like a sort of like a negative filter, right? They're going to specifically label cells that are in the blood, and then when they do their tissue extractions, they can essentially subtract that marker from the downstream analysis when they're looking. So, where am I going here? So they're basically doing a retroorbital injection, which is like behind the eye, injection of
anti-CD45.
Yeah, anti-CD45.
Yep.
So they're gonna label all of the cells that are circulating in the blood, and then very quickly after sack the mouse, and then do these extractions from a bunch of different tissues, and again do single cell analysis, do the clustering, do the labeling, etc. So they're looking at the blood, which they've labeled, and then they're pulling out brain, colon, fat, lung, and meninges here, and looking at the brain signatures. And as we said, the white fat had the most similar signature to the brain in this analysis. And so they quantify that in zooming in and out on my paper tablet is going slowly.
Well, real quickly, before we jump into
Yeah.
the quantification of that, I would say that, again, one of the things that they do well in this paper is they go back and forth between the mouse and the human a little bit. So before they get to the single cell, there is a little bit of flow analysis, just making sure that, that they do the initial tissue profiling by flow. But then they actually find a fresh post-mortem patient, basically, who's in that patient table. And they, to their credit, they do an analysis of lots of these different T cell populations. So at this point, they know that they've got this CXCR3 positive, interferon gamma positive, CXCR6 positive population that seems to be showing in the SFO of those animals. And indeed, you can find that population similarly across the animal, right? You can, similarly as they did the tissue screen across the mouse, you can also do that in human tissues. You can find a similar, like you said, axis, perhaps, similar T cell profiles across organs in a human versus in a mouse, which I do think is useful here. But then, of course, you get into the single cell, and basically just driving home what you said, looking for similarities between T cells in the white fat versus the SFO, right? And finding, again, similar signatures across those organs. Now, that's interesting. It's not really clear that means anything,
Right.
But that's fine. I think
so the next part of Figure 2,
yeah.
to me, this is one of the most interesting things. So
Yeah.
there is a model of chubby mice, fat mice.
Yes.
You basically give them a high fat diet. So standard chow is has a lot of fiber. It's it's plant based, obviously, but it's like a lot of fiber, carbs, protein, etc. There is a special chow that you can get that is high fat, and it makes these mice gain a lot of adipose tissue. Some of the early papers of this model, they sort of show pictures of the mice in the Nature paper, and it's like you've got these really obese mice, basically.
Yes.
Because they want to know, like, are T cells going to the fat, getting the signature, and then trafficking up to the SFO? And basically, they show that when you just give the mice the high-fat diet, those numbers don't go up that much in the SFO. But crucially, when you then have them fast, right? So you've got these mice that are fed in high-fat diet, so they're gaining a bunch of adipose tissue. And then you make them fast, you give them much lower calories. Now that fat is going to start to get recruited for energy consumption. It's going to get burned off. And then all of a sudden you see this huge spike in SFO T cells that have this sort of white fat-like signature.
Yep. And
And that, to me, is quite interesting from the perspective that I mentioned before of this enteroception, right? Like, what evolutionarily, why do you think when you are under fasting conditions, why would you have T cells, of all things, now traffic from your white fat where there shouldn't be much immune stimulation going on to the brain where there also shouldn't be that much immune stimulation going on? If you're thinking about the immune system strictly in terms of infectious disease, you're probably going to miss some of the point here.
yeah, I think you're absolutely right. And there's a point that maybe we'll come back to later, but I want to touch on now, which is they're going to eventually show that this is TCR specific. But they don't really talk about what the T cell receptor specific nature of this T cell engraftment in the white fat is. I think that is a fascinating question that needs to be followed up on, because I think they'll start to get at the end of this paper maybe why this would be relevant from a behavioral perspective rather than from an infectious disease perspective. But the targeting of this then gets left out, and I think that that's a really critical component that maybe we'll come back to later. So, anyway,
and so the last thing in Figure 2, it is implied you see a T cell signature in the fat. It goes up when these mice are fasting in the brain. The implication there is that cells are moving from the fat to the brain. Then they actually show this directly. So they have this cool photoconvertible mouse model. So you've got the T cells are expressing a green fluorescent protein. If you shine UV light on them, I wasn't totally clear. Are they just doing it like through? Is it just going through the skin? But they're basically treating the gonadal white fat with UV light. That's converting in a permanent way the green fluorescence to red fluorescence. And then they can say only T cells that have actually been exposed to this UV light are being tracked by the red fluorescent protein, and they can go look, actually, those are the cells that are increasing in population in the SFO. After fasting.
And I didn't go into the materials and methods around this, but I can tell you that a 2-photon microscope can get pretty deep into the tissue here. I mean, you can get, half a millimeter, or what it was like 500 microns is not so hard underneath. So it's shallow yet, but you can get underneath and you can start to hit stuff. So I imagine that's what they did, but certainly you can get light to penetrate. So then, and Kevin, I think that this is part of probably why you were interested in this paper. We get into Figure 3, which is fat is important. Adipose tissue is important when that diet gets changed. But another thing that happens when you start to change to a high-fat diet is the microbiome shifts dramatically. And so the question becomes, is some of this phenomenon, right, the mobilization of T cells from the fat into the brain, could that have anything to do with shifts in microbiome? And so they do an interesting kinetic experiment at the beginning, which is, well, if it's microbiome dependent, right, this axis that they're describing, you would expect a major transition from the weaning period across to the solid food period, right? So mice are weaning, or are receiving nutrition, right, from the mother right up until about week 3. Or so, right? In which case, or at which point, mice start eating solid food. And so you start to get an explosion of microbiome development, as you do in humans, as humans wean. And so the question is, do you see a corresponding increase to the number of T cells in the brain over that period? And the answer is absolutely you do. So Figure 3a basically shows you that at 2 weeks, when there is no solid food intake, there are no T cells in the brain. When you go to week 3, week 4, as those mice transition, that is when recruitment starts. And interestingly, if you take mice that are specifically raised in a germ-free environment versus a mouse that is raised in a specific pathogen-free environment, which is kind of the normal environment that we use our mice in, you basically find nothing in the germ-free mice. They essentially remove the T cells from the brain in those animals, or from the SFO, which is interesting. And so then the question is, can you overcome that by doing these add-back experiments that we love to do in germ-free mice? And it's never, to be honest with you, clear to me exactly how relevant these models are usually. But here, I think actually really helpful. So you're taking those germ-free mice and you're adding one specific bacteria. It's SFB. We study this all the time because I think it was one of the things that showed up early in the microbiome germ-free add-back experiments, basically showing that you could get Th17 differentiation.
Yeah, this bug, so it's segmented filamentous bacteria. It's a mouse bacterium. I don't think humans have it. This is the bug that came up in... There's sort of a thing. It was famous to us.
Yeah, it was.
I don't know if other people... it was like the time that we were in grad school, and it was Harvard labs, like the big Harvard labs that into this. But basically, there were, we've talked about this before, too. There were 2 labs that were studying Th17 cells, I think a model of MS, right? So these are SLE mice. Or not SLE mice, sorry. What do you call MS model in mice?
Yeah. Oh
EAE.
no. EAE.
EAE.
Yes. Yes.
So EAE mice, and like one lab was like, we do this thing. I think it was like MOG peptide injection or whatever. So we do this thing and we get tons of T cells. And this other lab was Th17 cells. The other lab was like, we do MOG injection. We don't get any Th17 cells. Turned out one lab had mice from Taconic. One lab had mice from Jackson. And I don't remember which was which, but one of the labs had SFB in their mouse colony. The other lab didn't because it came from a different facility. And the presence of that bug, which I think to this day it's still not cultured.
Yeah.
But that bug was basically solely responsible, or like was necessary and sufficient to get this Th17 injection. I don't know why it's relevant in this model. Honestly, like in most monocolonization experiments, like really a lot of the pathologies, so like germ-free mice are weird in a whole bunch of ways. Their immune systems are messed up, their behavior is different, like their diet is like they don't get as much nutrients. All kinds of things are messed up. If you just like a lot of those things can be overcome if you literally just feed them LPS, lipopolysaccharide. And so giving mice some microbial signal, giving them some toll-like receptor ligands, basically, like getting some amount of microbial stimulation is enough to recover, certainly not all, but a lot of the weirdness of germ-free mice you can sort of undo just by giving them a single bug. Frankly, SFB seems like a weird thing to give, and now that I'm talking about it, if they monocolonized with SFB, then they must have cultured it. I didn't actually look into that. Because how would you monocolonize if you couldn't actually culture it? So apparently that statement is wrong. But regardless, they're monocolonizing with this bug, and they're now recovering the migration of these SFO T cells.
Right. So then the question is, who are these T cells? Right? Are they random naive T cells? Are they clonal? Like, is it a single? If this is driven by a specific pathogen or a microbiome specific response, or is it the case that you're getting antigen specific T cells, right, as a component of this? Maybe it's not just sort of a generic T cell phenomenon. Maybe this is specific to a bug. So what do? You basically use that UV trick to sort out T cells from various locations, and you go through T cell receptor sequencing. This is the kind of receptor sequencing, adaptive immune repertoire work that is, it's thin. I would say there's not too much going on here. I think that we can generally summarize it as it is often useful to know how diverse a T cell receptor set is, a B cell receptor set is. The less diverse a receptor set is, the more clonally related those cells are, the less likely it is that you're just seeing sort of a generic T cell population versus a more specific T cell population. So I don't think we have to go into the details of what is shown in E through F, other than, yes, you can find clonal relationships between T cells across an animal, but interestingly, if you look in the brain, those T cells tends to be a little more clonal than a random T cell that you're going to find in the blood, which suggests to you that, it's probably specific to something, right? And they basically show in G that if you take...
Is that a good conclusion, or could it be that you're just saying, like, we're having small numbers of cells recruited stochastically and then expanding?
Sure, it could be. It could be.
Okay.
But I think that that's why they take it to G, which is a specific receptor situation, right? So basically you have these SMARTA T cells.
Before we do G, can I just make one nitpicky thing on E and F?
Please.
This is a figure critique thing. Advice. Do not use the same color scale to mean different things, especially in the same figure. Like, figure E has a color scale that represents rare to hyper expanded clonal groups, and then the exact same color scale from blue to red is then used to represent Jacquard distance in the different tissue populations, different clones. So it's annoying. Don't do it.
Yep.
They also they mean sort of opposite things, because like large Jacquard distances means there are less overlap, so less clonal in F, but in E, that color is used to meant more clonal. So anyway, sorry, that's just my my specific brand of OCD on figures. Made that figure very hard to read.
Color schemes mean something, and don't overcode. There's no need to put two visual variables on a single data variable. It is always tempting to do that because you want to like make data stand out sometimes. There's no reason that it has to be that way. So just I'll leave it there. So if you are thinking that perhaps this might be a receptor-specific response, one thing that you could do is you could check to see if a non-specific T cell is routinely left out of whatever this engraftment is, right? Because you'd expect that, if you have a high-frequency T cell, but it's not related to adipose tissue, it's not related to the microbiome, it's not related to anything, then that receptor should be excluded from the brain, right? And so to do that, they use
if it's just stochastic, what gets in there, you would expect a high-frequency T cell to get in at high frequency.
Yeah, exactly. It would be a representative group. And so if you go into a transgenic SMARTA mouse, and a SMARTA mouse is a mouse that has a CD4 T cell population that has a very specific T cell receptor to a component of the virus LCMV. And this is a virus that is relevant to mice. It's been used to study CD8 T cell responses especially a lot, but no relevance here, right? So this SMARTA T
no
cell
LCMV ever actually gets into this experiment.
Correct. And if it did, it would be tragic for the colony. So, so what you're doing here is you're basically saying, I'm going to take a mouse where 95% of its T cells are for this specific LCMV protein, and then I'm going to check to see what percentage of the T cells that I capture from different organs are actually specific to LCMV. And as you'd imagine, in a spleen where basically everything is naive, you'd expect that if the mouse is made 95% of these specific T cells, that about 95% of the cells in the spleen would be specific or would have that receptor. And that is what you see. About 90% of the T cells in the spleen are SMARTA positive. About 92% of them in the inguinal lymph nodes are SMARTA positive. But then interestingly, when you get into the liver, no longer the case, right? Now you seem to have the outgrowth of specific T cell populations that are actually not SMARTA related, and so SMARTA cells now only make up about 60% of the T cells in the liver. So certainly still some baseline engraftment, but now you're getting something more specific. But then you get to the brain where 90% now of the cells, the T cells that you're finding in the brain, do not have that SMARTA receptor. So what you're seeing here is that T cells are indeed.
I was really interested. Oh, sorry to interrupt, Matt. You froze for a moment to me.
Yeah. No, I was going to say basically what you're seeing here is that as these mice are developing, right, there is engraftment of T cells in the brain, but that T cell specificity seems to matter, and the cells that are obviously not specific for anything in the system, which are these SMARTA mice, are actually not the ones that are finding their way into the SFO of these animals, which I think is really interesting. And then if you basically
Can I just pause? I really wanted to know, based on that.
Yeah.
why didn't they look at the mesenteric lymph nodes? To me, that would have been interesting. So the mesenteric lymph nodes drain mesentery, the gut, right, the tissue surrounding the intestines. And given the link to microbiome and fat, and the liver is also like related to the digestive system, and I really like. What do you think they would have seen if they looked in the mesentery lymph nodes?
I don't know. It would be interesting to understand if there were biases. I think the liver is related to the digestive system is a statement that could have come out of a 1500s medical textbook. But
sure,
I like that.
Ask ChatGPT what it thinks. No, yes. I mean there's some interesting like anatomical stuff. I was recently talking to a GI physician talking about like portal vein blood, and so there's this like thing that basically like is directly between the intestine and the liver, and there's like a bunch of things that like portal vein blood is like different from other blood because it's from the gut but before it's been filtered through the liver. So what I'm saying is that there's close connection between these organs.
Sure. I didn't mean to dismiss
they're
you
physiologically linked, and it probably matters.
You're right,
asshole.
So, yep. So, one might guess then, right? Maybe there's a compensation mechanism. Maybe these SMARTA-specific T cells could get into the brain, but because there is another different T cell that competes for that microenvironment, maybe it's simply a low frequency event, right? And so the way that they did this to really confirm that those SMARTA T cells have no business in the brain because of an irrelevant receptor is they then take those SMARTA T cells and they put them on a RAG background. And so what they show basically is that if you have competent T cell differentiation outside of the SMARTA environment, you do get engraftment of those T cells that develop alongside the SMARTA cells. However, if you get rid of RAG outside of SMARTA, so now you have only SMARTA T cells, you basically lose T cells from the brain. So even if SMARTA T cells exist, and there are no other T cells to compete for this microenvironment, the SMARTA T cells, because of their receptor seemingly, will not engraft into that space in the brain, which tells you that there is a positive selection process going on in here. There are T cells that need to have some signal in order to go to the brain to begin with. And of course, they then show that with this T cell specific, essentially, to SFB, right? And I won't go into the details of this other than to say you can do the opposite experiment of everything we just did with SMARTA. You can say, I'm going to put in transgenic SFB T cells. I'm going to give mice SFB, and then I'm going to watch those cells that are SFB specific not only show up in the places where SFB is, but also then show up in the brain. And I think that's cool. That's an interesting experiment. It basically shows that just gut colonization and T cell responses in the gut are sufficient to drive those cells then to the brain, where they presumably have some biologic effect.
Yeah, and if SFB isn't around, they're not going to the brain.
They don't go to the brain.
So the exact same T cells.
Yep.
So they need some signal.
Yeah.
And just incidentally, they show in their little figure here, it's the Taconic mice that have the SFB, the Jackson mice that don't.
Right. Good deal. So
here, 15 years later, apparently these microbes have not crossed streams, which is interesting.
Yeah, they've gotten it figured out. Yep. So then they find a person who is willing to give 2 blood samples, a colon biopsy, a duodenum biopsy, which is small intestine, so you're like really up there, and then CSF, cerebral spinal fluid. Now I have to imagine that this person had a reason to be giving those biopsies outside of just normal research draw, because or this
You
is
want to hope that this wasn't
or
a
this is a
technician in the
technician
lab.
in that lab. Right? And if you were a technician in that lab, and this is you, we'd love to hear from you here at Audiommunity.
Podcast at autoimmunity.org.
Yeah, because we have opportunities for you here at the Hope Clinic at Emory.
At the very least, if it was somebody on this paper, I really hope it was Yoshida him or herself.
Yeah.
The first author of this paper,
Or the
that was
last.
doing that.
One of those two. Yeah. Somebody high up. If you were a middle author, don't do that.
Yeah.
Come on.
Demand more is what we're saying.
You best have been compensated. Anyway, so
In all seriousness, it is not ethical to provide authorship on manuscripts due to providing biological samples.
correct.
So both don't give samples for that reason, and also don't take samples for that reason.
Yeah. No, I'm sure, joking aside, I'm sure that this was all done ethically, but it is the case that oftentimes when you see these kinds of things, it's some person in the lab that's like, oh yeah, no, that doesn't sound like a big deal.
There is a very rich history of that in the gut microbiome fields. Like one of the earliest studies on infant microbiome is a guy just took all of the poop from his daughter for 2 years and sequenced it all.
That sounds right. I mean, the joke used to be the transplantation biologists early on, when they were trying to figure out MHC matching and all of that stuff, they would do it on themselves. They'd all have these little square scars on their forearms from trying to put transplants from each other, right? Anyway, scientists are weird folks. But one of the interesting things that they did was they took all of these samples and they asked the question, what makes a T cell, basically, without getting into details on this, what makes a T cell from the cerebral spinal fluid different than a T cell from any of these other locations, right? So you take all of these biopsies.
Say, just anatomically, the reason CSF is relevant here is because it is in the central nervous system. So it is in this ostensibly immune privileged space that the SFO and other components that they're talking about are part of. So it's beneath the meninges.
Right. And it's
in very gross terms.
Yeah, and it's surprising sort of to me that this actually works. I would not have anticipated that this experiment would work, but it does. So they basically pull T cells from these different environments. They do clonal trees like they did before. They find some specific clones that seem to show up in the cerebral spinal fluid of this human. And they basically ask the question, what's unique? And the answer seems to be that CXCR6 keeps showing up every time they look at the difference between T cells in the CSF versus T cells in other environments. CXCR6 keeps coming up, and that is something that came up before when they were looking at the single cell data from the mouse. And if you start taking a look at, where is it? If you start taking a look at sections of mouse brain and asking the question, is the ligand for CXCR6 in the brain, which is CXCL16, by the way, which I've never looked at. And I'm sort of a chemokine receptor person. This is
your T cell trafficking.
This is not where I've landed. But yeah, you get CXCL16 transcript if you do these little punch biopsies of serial sections specifically within the SFO. So basically what they've got here is indication that the T cells that you're gathering from the brain, from the CSF in this case, appear to be expressing CXCR6, and they appear to be enriched in spaces that are high in CXCL16. Interestingly, if you knock out... that's the next figure, isn't it? if you knock out CXCR6, you don't get the same. No, it's right there. So if you take a mouse basically that does not have CXCR6, so it doesn't have the homing receptor that you'd expect, you do get that decrease in engraftment in the brain, basically. So what they're basically establishing here is that there appears to be a CXCR6 CXCL16 interaction that appears to be critical in getting these things to the brain. So then all of this wraps up because it's a Nature paper with like, why, right? Why is all of this happening? Why would you expect a T cell under fasting conditions to find its way from the white fat up to the brain?
And more than that, a T cell that seems to, a family of T cells that seems to have antigen-specific responses to gut microbiome organisms.
Yeah.
Gut microbiome organisms are not hanging out in white fat, and they're certainly not hanging out in the brain. So, like, why?
Yeah. And so they start doing behavioral studies, basically. What happens if you don't have these T cells in the brain? What do they do? What do they not do? And the answer seems to be they change the way that they like food.
Yeah, this is bonkers. I love this.
And I'm actually gonna pass this off to you because I feel like this is right in your wheelhouse, right? And I think that this is a cool little finding that links up all of these different things together in a weird way that I would not have anticipated.
Yeah, I mean, I don't know that it's even worth going into the details. So they have these behavioral experiments where they can basically measure food-seeking behavior of animals that have been fasting. They have a bunch of different genetic tools, so they're showing that it's related to the interferon gamma signature of these Th1 T cells. It's related to the cells that are in the SFO. You know, if they knock out the homing receptor that you mentioned, if they knock out interferon gamma signaling, whatever, like these behaviors don't get modified. And basically, like when the mice are fasting, if they have the T cells, they are more prone to seek out. Is it a specific kind of food? No, I'm forgetting. It's several weeks ago that I read this in detail.
I don't remember reading that it was a specific kind of food.
No, it's just like food-seeking behavior, basically. Like,
Yep.
Are they trying to find food, and like are they willing to sort of go out of their way? In a normal mouse cage, the food just like sits above them, and it's like doesn't take any effort. So this is a behavioral test where they've got to like actually do some work to go look for food that they can smell. So it's interesting that these T cells are providing this behavioral cue. So what are the components we have? We have some kind of microbiome signal. Not clear that it's specific for any specific kind of microbe, just T cells that are responding to the microbiome. These T cells are navigating to fat, where if you've got a ton of fat, if you have high caloric density, these T cells are not trafficked into the brain, but during periods of fasting or when that fat is being burned, these T cells are migrating to the brain, and they are triggering somehow via interferon gamma signaling. They are triggering behavioral responses that are increasing food seeking. Why? Like what? Like I still don't have a great I don't have a great explanation for this. There's clearly like when you are fasting, you should want to look for food. Like that makes sense, right? And there's a bunch of hormones that are related to, like, if you are, if you have low insulin, for example, because you have like low blood sugar, like this triggers hormonal responses that are going to increase your desire for food. And it also makes sense, like the immune system has a lot of like signaling possibilities. via cytokines and all kinds of things. The immune system is interrogating lots of different organs through circulation, so it kind of makes sense that you would link these things together. I can't think of an explanation for why you would need microbial signals and why it makes sense to have this additional signal over what we already know about hunger leading to food seeking. Like that link, I mean, it's fascinating. I think this is like, it's bonkers that this exists. Like, you know, you could imagine this kind of thing happening for like specific nutrients, like different ways of triggering the brain to, but yeah,
Does it feel like a parasitic hack to you? It feels like a parasitic hack to me almost, where like you had these responses in the gut. You've got a microbiome that is trying to regulate, right? And at some level, in order to keep that microbiome healthy, it would be useful to have a feedback mechanism that would find its way into the behavioral
Yeah, but this
realm.
isn't a microbial effector. Like if you found a microbe that produced some like effector through type 3 secretion that induced something, like then I would buy that explanation, right? But this seems like this doesn't seem specific. Like it's not SFB.
Yeah.
That has evolved some mechanism to get mice to eat more.
Right.
Like, this is a physiological, evolved thing in the mouse. And the human, apparently.
Yeah.
You know, given their data, I'm convinced by that. So...
But what if it started that way? Because, like, it feels like a weird duct tape workaround, honestly. And it makes me wonder if, like, an early co-evolved pathogen, or not pathogen, but, you know, if there was that kind of initial effect where you say, oh, well, actually, you know, the survival increases if the microbe is capable of driving food-seeking behavior in the and then it just becomes a generic mechanism that sort of winds its way into... Anyway, I don't have a good explanation for why this happens. I guess that's the point. And so I've been trying to come up with good ways to evolutionarily explain why this might happen this way, and I'm struggling for it.
Yeah, I honestly, like if it was the case, I mean, so one thing to say is that animals always have a microbiome, except in some weird mouse facilities, right? Germ-free mouse facilities are a very unusual circumstance. So it could be the case that this is just a signal, like there's some evolutionary mechanism where T cells that are patrolling the gut, if they don't have anything better to do, because these aren't pathogens that they're responding to, they're not trying to clear it. So, like, let's say you have a T cell, it makes a T cell receptor to a microbial product, not triggered during infection, but you've got these T cells, they're activated, they're hanging around, they're not needed for clearing infection. Like, maybe we're going to recruit those to provide some signal that we need. I could sort of see that happening. It doesn't make sense that it's like what you were talking about, like it's like the pathogen, or it's like a bug inducing that has evolved to induce this. But yeah, it is a weird hack.
I don't get it.
Seemingly evolutionary hack, which is not unusual in evolution.
Sure.
Right? Like, we use what we have.
Yeah. And sometimes it gets plugged in weird ways, and you're like, that's probably not the most efficient way that could have been done, but, you know, it seems to be working. My favorite part of the explanation for this in this figure is actually the last cartoon, where it's like something happens in the gut, something happens in the white fat, they go into the blood vessels, there's stuff in the brain now, and... behavior happens.
Yeah.
Yep.
The thing I'm making on if you are watching this on YouTube, the figure is a little stick figure that like has little things shooting out of his brain.
Right. Right.
Yeah. No, it's wild. I think it's fascinating.
It's cool.
Like I said, I'm very interested in this. There are clearly a lot of links between the gut and the brain. There are clearly a lot of links that are mediated by the immune system.
And we've
most of the way that I am used to thinking about that is like T cells responding to the state of the microbiome are secreting cytokines in the gut that are then circulating, and the brain is responding to those circulating cytokines, like sort of global state. This idea that you have T cells that are literally like carrying messages from the gut to fat to the brain, like that's wild.
Yeah,
it's wild.
The nature of those messages, I think, is what's confusing to me. But one of the things that it brings up is a discussion that we've been having for a decade now, which is this interesting phenomenon where parasitic infection or exposure to parasites in the gut drives T cell response and polarization that seems to have direct impact on the brain. And we've talked about the parasite and helminth versus MS studies in the past. Go listen to those podcasts because those are really interesting. But basically what that comes down to is that dramatic changes in T cell populations in the gut can have really dramatic changes to the T cell populations in the brain, specifically those that drive autoimmunity. And you always think about that as sort of like a one-off, right? Like there's this weird phenomenon that's happening, T cells that are poorly trained. But as you integrate these systems into that thinking, it's actually not the one-off, right? It's that you've got a modified, potentially resident population of T cells, and you've got this constant access where T cells are constantly trafficking just homeostatically between the gut and the fat and the brain, right? And then you add the helminth component on top of that and the autoimmune component on top of that. And all of a sudden you're talking about this much more dynamic system that isn't like, oh, these T cells are here by accident and they're bad, so let's fix them in the gut. Now you're talking about realigning a full homeostatic process that might be trafficking. And I think it broadens the scope of how I think about all of this, how it could work.
Here's an idea that just made me think about.
Yeah.
So helminths drive classically Th2 responses.
Right.
What they're talking about here is Th1 responses, interferon gamma triggering this. Classically, Th2 and Th1 are opposed. What if, different from your idea that these are bugs that are inducing to feed themselves, what if this is an immune response, like a sort of backdoor way of saying, if you have a helminth infection and you've skewed a ton of stuff to Th2, maybe we don't want to be seeking more food. Maybe we want to starve a little bit. Reduce the amount of gut intake, rely more on our adipose tissue to try to get rid of that helminth infection. It's indirect. Again, it's like a weird hack. It's backwards. Like, you would think that instead IL-4 would suppress appetite or something, like if that was your goal. But like,
I think, it's as good an avenue as investigation as I can come up with. I really like the idea, and we've talked about this before in a number of contexts. I really like the idea of behavioral immunology in general, and how molecular systems drive behaviors that then help you avoid infection or help you deal with infection. I mean, the fever behaviors we always talk about, right? IL-6 changes the set point in your body for what your internal temperature should be. But that is actually accomplished because then your brain tells you to shiver and get under a blanket, right? Which is interesting, right? It's not that it happens by magic. It's that you're driving behaviors that change the way that your body interacts with the outside world and the interior environment. And I like the idea that changing polarization in the brain might lead to different behaviors that would change the way you interact with outside pathogens. But cool. Exactly. Behaviors.
Yep. Anyway, I saw this on Blue Sky. I saw like, what the hell's a subfornical organ?
Yeah.
But T cells, and then I saw, gut microbiome. So this just like hits all of my high points. We've got location.
Yep.
We've got gut-brain immune axis. We've got T cells in places where you wouldn't expect. Cool paper.
Yeah, it was. I liked it.
So I think with that, we can wrap up. My computer is at 21%. I need to remember to bring my charger with me because I'm plugging into a dock at work. I don't. Anyway, this has been Audiommunity, recorded on July 10th, 2025. If you like what we're doing here, if you find these conversations valuable, you can support us on Patreon, patreon.com/Audiommunity, AUDIOMMUNITY.org. You can subscribe to the podcast via RSS feed that can be found at our website, autoimmunity.org. And we've got all of our previous episodes accessible there. You can also like and subscribe on iTunes or whatever podcatching service you're using. If you do that, please leave us a rating and review. The most important thing you can do, really, like even more than subscribing or supporting us on Patreon, is share us with a friend. Send a link to this YouTube video, to this audio file. Let somebody else know that this is a conversation that they might like to hear. We'd really love to increase our reach. We don't currently have a good contact solution. I'm thinking that I'm going to use a little bit of our Patreon money to just get an email inbox for the website. So I'll put that up as contact info. Again, autoimmunity.org. Maybe I'll do that before I actually post this podcast. So if you're listening, you can find our email address on autoimmunity.org. It'll probably be something like contact@autoimmunity.org. And music at the beginning and the end of the show was composed by Rachel Reinick. Hopefully Kate will be joining us next time. And talk soon.
Thanks, y'all.
Sometimes